5%. That is the share of the global population that carries hazel eyes. Not brown, not blue. That shifting, layered, impossible to name color that looks green in morning light and gold by afternoon and something closer to amber the moment a cloud passes overhead. For most of the 20th century, geneticists had no category for it. It was a rounding error, an intermediate, a blending artifact that happened when dominant and recessive alleles didn't sort cleanly. They gave it a bin and moved on. That was the mistake. Because when researchers began extracting DNA from ancient skeletal remains buried beneath the limestone hills of the Levant, they didn't find noise. They found a signal. A precise, concentrated, geographically specific signal pointing to one of the most dramatic population mergers in human prehistory. And the color sitting at the center of that signal wasn't blue, which everyone had studied. It wasn't brown, which everyone had assumed. It was hazel, the trait no one had bothered to map. The trait that, once mapped, rewrote what geneticists thought they knew about the ancient Middle East, about migration, and about what is written inside the iris of anyone alive today who has ever struggled to name their own eye color. The genetics of eye color was supposed to be one of the easy ones. Simple inheritance, clean ratios, the kind of trait introductory biology courses used as a teaching tool because the math worked out so neatly on paper. Brown was dominant, blue was recessive. 2 alleles, 1 gene, a Punnett square on the back of a textbook, and you could predict a child's eye color before they were born. That was the story for most of the 20th century, and it was wrong in almost every detail. The reality was far more complicated. Eye color is not controlled by 1 gene. It is controlled by at least 16, interacting through epistasis, incomplete dominance, and long-range regulatory feedback loops that stretch across thousands of base pairs on chromosome 15 alone. At the center of that region sits a gene called OCA2, which manages how much melanin your iris produces. Next to it sits HERC2, which contains a regulatory enhancer that acts as a dimmer switch for OCA2. When that enhancer carries the ancestral variant, melanin flows freely. The iris goes brown. When it carries a derived mutation, the switch goes quiet, melanin drops, and the iris lightens toward blue. That much, the textbooks eventually corrected. But the model still couldn't explain hazel. It couldn't explain how 2 brown-eyed parents produce a hazel-eyed child. It couldn't explain why the same iris looks green under fluorescent light and gold in direct sun. The model made hazel genetically invisible, and invisible things don't get studied. Nobody plotted hazel on a map. That was the oversight that held the field back for decades. Researchers were sequencing European cohorts, building Danish biobanks, running Scandinavian registries, and treating hazel as a minor footnote in studies designed to explain the blue-eye founder mutation. Nobody stopped to ask a simple geographic question. If hazel were just statistical noise, it should distribute roughly evenly across populations that carry both brown and light eye alleles. It doesn't. 5% of the global population carries hazel eyes. That is the baseline. But when you break that figure down by region, the baseline stops making sense. In Lebanon, surveys suggest hazel eye frequency may reach between 15 and 25%. In Syria, estimates range from 12 to 20. In Turkey, 10 to 18. In Iran, 8 to 15. Every one of those figures is 2 to 5 times the global average, concentrated in a geographic arc stretching from the eastern Mediterranean coast through northern Mesopotamia into the foothills of the Zagros Mountains. This is not a minor elevation. This is not sampling error. A trait appearing at 5 times its global average in a specific corridor is not noise. It is a signal. And the corridor it traces, from the Lebanese coast through the Syrian interior into the Iranian highlands, is not a random slice of the map. It is the exact region where 3 of the most genetically distinct ancient human populations in history came into contact. The numbers were pointing somewhere specific. It just took the right evidence to follow them there. When a signal this strong goes undetected for this long, the failure is never accidental. It is structural. There were 3 compounding reasons science missed the hazel concentration in the Middle East, and each one made the next harder to correct. The first was the model. Teaching eye color as a single gene, 2 allele system didn't just oversimplify the biology, it actively erased hazel as a category worth investigating. If brown is dominant and blue is recessive, then hazel is just brown that didn't finish. It becomes a gradation, not a phenomenon. Gradations don't get their own research programs. They get folded into the nearest clean category and forgotten. The Punnett square was not just wrong, it was wrong in a way that made asking better questions feel unnecessary. The second was the sampling. The landmark studies on eye color genetics drew their subjects almost entirely from Northern European populations. When Middle Eastern individuals appeared in the data at all, they were used as brown-eyed controls, baselines. Nobody sequenced the Lebanese grandmother with the gold ring around her pupils. Nobody asked what was happening inside those supposedly homogeneous, dark-eyed populations. The question was never posed because the framework didn't suggest it needed to be. The third reason was the deepest. There was no ancient DNA from the right time and place. Without genomes from the Chalcolithic Levant, researchers could see the pattern in living populations, but had no way to trace it backward. They could read the result, they just couldn't find the cause. That changed because of a cave. Pekian Cave sits in the limestone hills of Upper Galilee in northern Israel. It was not discovered by an archaeological expedition. It was discovered by accident in 1995 when construction workers broke through a natural rock seal and found themselves looking into a darkness sealed for roughly 6,500 years. Archaeologist Dina Shalem led the excavation. What her team uncovered was unlike anything previously documented in the region. Inside, approximately 600 individuals had been buried during the late Chalcolithic period, around 4,500 BCE. The dead had been placed inside ceramic ossuaries, ornate containers shaped into stylized human faces, some standing over a meter tall, with wide staring eyes and prominent noses. The craftsmanship was not local. The metalwork included copper objects traced to technology from Eastern Turkey or the Caucasus. The pottery matched nothing that came before it in the Levant and nothing that came after. This culture appeared, flourished for roughly 600 years, and vanished. For 3 decades, that gap haunted every researcher who studied the site. The people who built those ossuaries, who carried that copper technology into the Galilean hills, who buried their dead with such precision behind a limestone seal, had left no clear ancestor in the archaeological record. The question of where they came from sat unanswered until a team with the right tools finally asked it of the bones themselves. In 2018, a team led by Edwin Harney and Professor David Reich at Harvard University, working alongside Dr. Ela May and Professor Israel Hershkovitz from Tel Aviv University, extracted bone powder from 48 skeletal remains at Pechiin. From 22 individuals, they reconstructed viable genomes. The cave's cool limestone interior had acted like a biological time capsule, sealing the DNA against the degradation that destroys most ancient genetic material long before anyone thinks to look for it. What those genomes revealed was not what anyone had predicted. The Pechian people were not a local population. Their ancestry broke into 3 distinct streams. Roughly 57% matched Levantine Neolithic populations, descendants of Natufian hunter-gatherers who had lived in the region for millennia. But 26% matched Anatolian Neolithic populations from what is now Turkey, and 17% came from Iranian Chalcolithic groups in the Zagros highlands. 3 populations that earlier studies had shown were as genetically differentiated from one another as modern Europeans are, from modern East Asians had fused into a single community, within what appeared to be a single generation's worth of burials inside one cave in the Galilean hills. The Chalcolithic Levant was not a backwater absorbing outside influence slowly. It was a collision point. And the collision left its mark not just in the pottery and the copper, it left it in the DNA, including the DNA that controls what color the iris becomes. Among the Pechian genomes, 49% of individuals carried the allele associated with lighter eye color. That variant had been completely absent in earlier Levantine populations. It arrived with the northern migrants, carried south through Anatolia by people who brought copper technology, distinctive pottery, and something written into their DNA that the Levant had never seen before. But the blue eye switch was only half the story. Hazel does not work the same way as blue. The genetic architecture that produces blue eyes requires a near-complete shutdown of OCA2 expression. Melanin collapses. The iris loses almost all its pigment. Hazel requires something more precise, a moderate dimming, not a shutdown. The variant responsible is a missense mutation called RS180407, a single nucleotide change in exon 13 of OCA2 that swaps one amino acid for another at position 419. That substitution doesn't silence melanin production. it reduces it, just enough to shift the iris out of solid brown and into the layered, shifting territory of green, gold, and amber that resists every simple category the old model tried to impose. The Natufians contributed the brown baseline. The Anatolian farmers carried light pigmentation variants shaped by thousands of years of independent evolution. The Zagros Highlanders added their own distinct genetic layer. And where those three streams converged, the Hazel signature emerged at concentrations found almost nowhere else on Earth. Before the final piece, if you want to trace where Hazel fits into your own bloodline, I put a full 3-volume PDF bundle over at the Origin DNA website. It shows you how to read your raw data and find matches beyond what Ancestry or 23andMe tell you. Link is in the description. But stay right here, because what comes next is the part most people completely miss. Once RS1800407 reduces OCA2 expression and pulls melanin into a moderate range between full brown saturation and near absence, something else takes over. The biology steps back and physics steps in. And physics is what makes Hazel unlike any other eye color on the spectrum. The iris is built from two main layers. The back layer, the pigmented epithelium, is densely packed with dark melanin regardless of eye color. Every human has it. The front layer, the stroma, is where the difference lives. In brown eyes, the stroma is also heavily pigmented. Light enters, gets absorbed, and very little scatters back. In blue eyes, the stroma carries almost no melanin. Short wavelengths scatter backward through near empty tissue, and the eye appears blue through the same mechanism that makes the sky blue. No blue pigment exists in any human iris. In hazel eyes, the stroma sits in between. Melanin is present but unevenly distributed, concentrating near the pupil and thinning toward the outer rim. That gradient creates the characteristic sunburst pattern. In the low melanin zones, shorter wavelengths scatter backward. Blue mixes with residual yellow-brown pigment from pheomelanin. The eye appears green. Shift the angle, change the light source, and the ratio tips the other direction. Green becomes gold. Gold deepens toward amber. No pigment has moved. Only the light has shifted, and the eye has answered differently. When hazel eyes appear in Middle Eastern populations at 2 to 5 times the global average, the instinct is to reach for a recent explanation. Crusader contact, Ottoman-era admixture, European gene flow over the last few centuries. It is a reasonable instinct. It is also wrong, and the ancient DNA makes that clear in a way that living population studies never could. The RS1800407 allele does not behave like a recent import. Its geographic distribution does not follow the routes of medieval European armies or Ottoman networks. It concentrates in the Levantine interior, in the Syrian highlands, in the Iranian foothills, in exactly the regions least touched by sustained European settlement. It is absent in East Asia. It is absent in sub-Saharan Africa. Its fingerprint maps precisely onto the zones where Anatolian, Caucasian, and Iranian Chalcolithic lineages overlapped with local Levantine populations during and after the Chalcolithic period. That is not the pattern of recent contact. That is the pattern of ancient admixture. And critically, RS1800407 is not a diluted version of the blue eye switch. It is a separate genetic event, operating through a different molecular mechanism, affecting a different position in the OCA, 2 protein, producing a different phenotype. Hazel is not blue that didn't fully express. It is not brown that partially faded. It is the product of a distinct mutation carried into the Levant by a distinct migration. The textbook that lumped it with brown wasn't just imprecise. It was erasing a 6,000-year-old story written in a single nucleotide. Here is where the story takes an unexpected turn. If hazel eyes are this old, this geographically concentrated, and this genetically distinct, the natural assumption is that something must have selected for them. Some survival advantage, some environmental pressure that pushed the trait forward through 6,000 years of invasion and population replacement. That assumption turns out to be incorrect, and the incorrectness is itself the most revealing finding of all. The blue eye haplotype shows strong signatures of positive natural selection in European populations. The HERC2 region displays extended haplotype homozygosity, a signature that appears when a variant spreads faster than random drift alone can explain. Something actively favored blue eyes in Northern Europe. The leading hypotheses involve light sensitivity at high latitudes, melatonin regulation during dim winters, or sexual selection. The debate continues. But the selection signal is unambiguous. The Hazel variant shows none of this. A study genotyping over 3,400 individuals from 72 populations across 21 markers in the OCA2 and HERC2 region found that RS1800407 did not reach significance for extended haplotype homozygosity. When researchers modeled selection pressure on Hazel-linked alleles, the signal came back too modest to drive the trait to dominance. Not enough to explain its persistence through 6 millennia of demographic disruption. Hazel was never pushed by evolution. It was carried by people. And that distinction changes everything about what its survival means. Consider what the Middle East absorbed between 4,500 BCE and today. Assyrian conquest, Persian administration, Greek colonization, Roman occupation, the Arab expansion of the 7th century, Crusader kingdoms carved out and dismantled within 200 years, Ottoman rule stretching across 400 more, wave after wave of military and demographic force washing across the same narrow corridor where the Pechean people once buried their dead behind a limestone seal. And through all of it, the Hazel signature held, not because it was useful, not because it conferred any advantage selection could amplify, but because the 3 populations that created it never fully replaced one another. Each conquest reshuffled the surface of the region without erasing the underlying genetic architecture those 3 streams had built together. Empires can replace languages, religions, and political structures. They cannot fully replace the allele frequencies sitting quietly inside a population's genome. Today, in Beirut, in Istanbul, in Tehran, Hazel eyes still appear at 5 times the global average. When light catches an iris on a busy street and the color shifts from green to gold in the time it takes someone to turn their head, that optical event is not incidental. It is RS1800407 doing exactly what it has done for 6,000 years, dimming melanin to a precise threshold, letting physics scatter the remaining light into colors that resist every category. The empires came and went. The allele remained. An estimated 400 million people alive today carry Hazel eyes. Most have spent their lives struggling to answer a simple question on a form. Green? Brown? Both? Neither? The uncertainty feels personal, almost arbitrary. It is neither. It is ancient. The arc from Beirut to the Zagros Mountains still holds the highest concentration of Hazel eyes on Earth, 5 times the global average. In the same corridor, where 3 branches of humanity converged 6,000 years ago and never fully separated again, the Natufians brought the melanin baseline. The Anatolian farmers brought light pigmentation alleles shaped by thousands of years of independent evolution. The Zagros Highlanders contributed a genetic layer researchers are still working to fully map. And where those 3 rivers of ancestry met, physics and biology combined to produce something neither population could have generated alone. No empire erased it, no migration replaced it. It drifted forward on demography's inertia, carried passively through every upheaval the region absorbed, until it arrived here, readable in a single nucleotide, visible in an iris, present in the eyes of anyone who has ever watched their own reflection shift color in changing light and wondered why. The answer was always there. It just took 6,000 years and the right cave to find it. The history is on the map, but it's also in your code. To unlock it, check out the Origin DNA at the link below. It's a 3-volume PDF guide bundle that walks you through reading your raw data, mastering your matches, and tracing your deepest origins. Don't settle for surface-level test results. Click the link below and get your bundle.